Bismillah ar-Rahman ar-Rahim. Good evening, everyone. This is Asma Yamani, a student at King Fahd University for Petroleum and Minerals. Today I'm going to be presenting a sequence-based approach to classify activation-inhibition relationship of protein-protein interaction using machine learning techniques. Protein-protein interaction networks are graphs where every protein is represented as a node, with an edge signaling that two proteins interact with each other. They're important for inferring signaling pathways. Signaling pathways play important roles in understanding the underlying mechanism of cell growth, proliferation, and other biological phenomena. They're also important for understanding disease mechanisms and drug development. With the use of high-throughput mechanisms for PPI detection, an abundant amount of PPIs have been discovered. However, these mechanisms tell you if two proteins bind with each other, but they don't tell you the basic information you need in order to reconstruct signaling pathways. In order to reconstruct signaling pathways within a PPI network, you should have knowledge of the relationship, naming activation or inhibition, and the signal flow direction, whether it was upstream or downstream. Previous work done on PPI relationship prediction can be categorized into two ways: phenotype-based methods and Go-based methods. Phenotype-based method used RNA interference screening to identify phenotypes resulting from interactions. Protein resulting in similar phenotypes would have activation relationship, they would have inhibition otherwise. The limitation of this method is that it needs phenotype data, which may be impractical for larger PPI networks. Also, RNAi screening is expensive and infeasible for larger PPI networks. The second category is work that derives the features from gene ontology. The first work that was done used Go terms as their features, and they hypothesized that if two proteins have similar Go terms or similar Go annotation, then they would interact positively, meaning activation. Otherwise, they would have inhibition. The second work that was done used Go relations for their features. They were able to predict both relationship and direction with accuracies reaching 89%. The limitation of this study is that it needs Go terms or Go relations, which may not be available for all PPIs. So here we propose a sequence-based method that relies only on the sequences of the two proteins interacting in order to predict the relationship. So we have four different status. Either the relationship is activation or inhibition, or both of them, activation and inhibition, or none of them, meaning other. To make the classification, we would run the sequence into both models. If the activation model and the inhibition model came out as positive, then we would have activation and inhibition relationship. If both of them came back as negative or zero, then we would have the other status. If either one of them was positive, then the relationship is activation or the relationship is inhibition. In order to build our model, we downloaded the Homo sapien PPI network dataset, which includes more than 3 million PPIs from 84,000 different proteins. The database includes protein-protein interactions along with their type, action, direction, and confidence score. To build the positive subset for the model, we took the PPIs that their type is labeled as inhibition or activation, with a score greater than 700 to only take high-quality PPIs, and then we divided them whether the action was inhibition or activation and inhibition. Then we removed the duplicates, and then from inhibition, we removed PPIs that also have activation action, and from the activation subset, we removed PPIs that also have inhibition action. Then, to generate the negative subset, we took the PPIs that are labeled as reaction, expression, PT mode, binding, or catalyst. We also took the PPIs that have scores higher than 700, and then we removed the duplicates. We removed PPIs that do have activation or inhibition in their action, meaning as the relationship. And then at the end, we have our negative subset. After that, We combined the subsets that resulted in order to build a balanced dataset for our activation model, to train our activation model, and another balanced dataset to build and train our inhibition model. As for training and testing, and since we only are using data from the string database, we took 30% of the data randomly out as a holdout test set. We're not going to use it in building the model or training the model. So all the results that you're going to see, except for special occasions, are going to be from the 70%, the rest of the data. And for all the hyperparameter optimization and for building the model and testing, we use tenfold cross-validation on this portion of the data. After our dataset is now ready, we move to feature representation, where we used Word2Vec, a popular natural language processing technique, in order to generate the vector embedding in order to derive features from the sequence. This technique has been used before in bioinformatics. And there are 3 ways to make the tokenization for the sequence. The first one is the single way, where we would generate embedding vector for each amino acid. The second way is the overlap 3-gram, where we would divide the protein sequence into 3 window-sized overlapped and generate embedding vector for each of every overlap 3-gram. The third way is the non-overlap 3-gram, where we would divide the protein sequence into 3 window sizes and then shift the 3 window size to generate 3 new sequences and generate embedding vector for every non-overlap 3-gram. To generate these 3 feature representations, first we took the FASTA file that contains all the protein sequences. We manually did the overlap sequences of this file, and then we generated the corpus and trained the model with a library called BioVic. It's available on GitHub. After training the model and generating the feature representations for both proteins, for the protein-protein interaction, we concatenate all vectors and attach them to the target. And this just shows an example how would one row of our training or testing file look like for one PPI. We would first have the vectors for representation vectors for protein A, and then protein B, and then the target. In order to decide which tokenization method would work best for us, we trained the model using logistic regression and ran the prediction for the inhibition relationship, since it was the lighter file. After that, as we can see, we have the best performance with the non-overlap 3-gram. So for all our upcoming experiments, we're only going to use the non-overlap 3-gram for our features. We proceeded after that for making the predictions with the 3 different machine learning techniques. We used the implementation provided by scikit-learn. We used logistic regression, support vector machine, SVM, and random forest. We did run grid search CV and random search CV for hyperparameter search to find the optimum parameters, and we did over 900 fittings to find the optimum parameters. As we see from the result of predicting the relationship experiment, that a random forest performed the best, then SVM, and then logistic regression in terms of accuracy and in terms of F-score. However, since we didn't reach to the 90% accuracy that we hoped for, we decided to go forward and use CNN-LSTM neural network to make the predictions. Our CNN-LSTM architecture is going to be composed of 2 one-dimensional convolution layers and then an LSTM. Our activation method that we're going to use at the end is the sigmoid function. In order to avoid overfitting, we ran a hyperparameter search for the dropout and the number of epochs. As we can notice, it's different for each model. This can be attributed to the different number of instances each dataset has. As we can see here, all the scores, accuracy, precision, recall, F1 were very close to 0.9 or above that. And this is what we were hoping for. Even for the accuracy score of the whole data set, and the accuracy for activation was 92% and for inhibition was 90%. As we can see here, CNN-LSTM does outperform previous machine learning techniques that we used, especially in terms of accuracy and the F1 score. We're going to look now at the last two experiments, which are predicting the presence or absence of both relationships. Meaning that, given a PPI, if both activation or inhibition exist, or both activation and inhibition do not exist. Not activation nor inhibition exist. So what we did is we took out the holdout test sets for activation and inhibition. Those holdout test sets were sure they were not involved in building or optimizing the model. And then we intersect them with the subset, not activation nor inhibition, where we are sure that both relationships do not exist. The intersection would give us the subset that we did the evaluation on. We did the same thing for testing the presence of both relationships, where we took out the holdout test sets and we intersect them with activation and inhibition subset. Looking at the results, we can see how tricky these cases are. For predicting the absence of both relationships, we can see that both SVM and logistic regression did below 75% accuracy. As random forest and CNN-LSTM did pretty well with 90% to 92% accuracy. The harder problem, as we can see here, is detecting both activation and inhibition at the same time. Although, for example, SVM did well above 75% when detecting activation or detecting inhibition, the combination of that resulted in accuracy of about 65%. And random forest logistic regression had even lower accuracies. CNN-LSTM did pretty well, which shows about 83%, which indicates how powerful deep learning is and the architecture chosen. When comparing with the previous work, we'd like to compare first with the work done in 2016 that used the Go terms, since we were using the same database. We can see here that our results using CNN-LSTM did outperform the results from 2016. When it comes to the study that used the Go relations, we're using different databases, so the comparison may not be fair, but we did almost equal the same when it came to detecting activation, and when it came to detecting inhibition, our method did perform better. We can notice here that neither studies did report the result of detecting activation and inhibition, and we did slightly better than the first work when detecting activation, the absence of both relationships. And finally, the takeaway here is that the sequences of two interacting proteins can be sufficient to predict the relationship of the interaction when utilizing powerful feature representation techniques such as word-to-vec combined with deep learning. I'd like to thank Dr. Iman Budlewa for her guidance and encouragement throughout the journey of the project. Also thanks to Hissa, Randa, Reem for their help and valuable discussion. Thank you all for listening.